Good afternoon, everyone. Welcome to the Wednesday afternoon lecture. As we anticipate some sort of a snowstorm, I'm glad you all are here, both in the Missouri auditorium and those who are watching on the web, probably in great numbers, because we really have a very special speaker today, Dr. Christine Seidman, known to all of us who know her well as a friend as Cricket. She is the Thomas W. Smith Professor in Medicine and Genetics at Harvard Medical School. She's also the Genetics Director at Brigham and Women's Cardiovascular Genetics Center. She's an investigator in the Howard Hughes Medical Institute and a Senior Associate at the Broad Institute of Harvard and MIT. She did her undergraduate work at Harvard, got her MD at George Washington University, did residency at Johns Hopkins, where I gather from reading a little bit of interviews with her, she was influenced particularly by one Victor McKusick in terms of how to bring science and medicine together in interesting ways. And boy, has she lived up to that exhortation by Dr. McKusick. Subsequent to that, she did a research fellowship in medicine in the cardiac unit at Harvard, and she has been continuously, since that time, in Boston, as I mentioned now, as professor in Brigham and Women's Hospital and at Harvard Medical School. She, more than anyone else, has really defined the cardiovascular genetics field as it relates to particularly cardiac failure, but also to cardiac developmental abnormalities, and taken a field which was largely descriptive and caused it to become intensely and interestingly molecular, with many consequences, both for diagnosis and also for interventions to assist people whose families are afflicted by these disorders. So it is a great pleasure indeed to have the opportunity to have her here for this Wednesday afternoon lecture, and I would ask you please to join me in welcoming Cricket Seidman.
Dr. Collins, thank you so very, very much, and to all of you who stood with me against this nasty storm that is coming from the south and hopefully not going all the way to Boston. I really appreciate you coming this afternoon. It's an incredible honor to be here. Some of you may know that I spent a very important year in my life after leaving Johns Hopkins in NICHD Building 6 when it was a little bitty small place, and it's always fun to see how much this has grown, and particularly fun to see how many incredibly talented, bright, smart, and often female scientists are here today. So thank you for having me come back. What I'd like to do in the next 45 minutes or so is to tell you a little bit about how we're really using genetics to understand causes of heart diseases that occur in rare familial forms and also in populations. And equally important, how we are seeing the complexity of using this new diagnostic technology to try and understand how similar phenotypes that arise in a single individual might be interpreted using genome sequencing. And finally, in the last little bit, I hope to be able to tell you a little bit about how there are new strategies, and I'm going to tell you about only one, but many are still under development, to try and use that genetic information to actually use and create genetics therapies. And the study unit, if you will, is going to be, as Dr. Collins said, on cardiomyopathies, something that I've been interested in for a very long time. I will remind all of you that this is by far and away the most beautiful organ in your body. Not only does it function 24/7, but for those of you with a stethoscope, you know that it sings to you. And if ever you're unsure of whether you want to continue in your path of medicine or not, just go listen to a heart with a great murmur, and I'm sure you'll come along to the cardiac side of things. Unfortunately, faced with the many insults that we throw at our hearts and indeed our entire bodies, this organ often undergoes a lot of remodeling. But unlike many other different tissues, it really takes one of two distinct courses. Either it results in cardiac hypertrophy, where the walls of the normal left ventricle become profoundly thickened and the cavity size is diminished, and this can be a genetic condition or a spontaneously occurring condition, or the walls become significantly thinned and the intravascular volume is actually increased, sort of as an overextended balloon. For years, we've known that lots of secondary causes of this might be due to hypertension, valvular heart disease, or myocardial ischemia and viral myocarditis. But in many individuals, we've also known that these arise spontaneously and often in young people, which represents footprints of genetic diseases. Over the past several decades, in fact, there have been the discovery of many different genes that can give rise to each of these morphologies, and we call these intrinsic cardiomyopathies. Those that cause hypertrophic remodeling are shown here in the upper left. The most important of these, based on their prevalence, are mutations in the sarcomere genes that encode the contractile unit of the heart. Similarly, we've identified many other genes that can give rise to primary ventricular dilatation. Again, you'll notice sarcomere genes do this as well, but one in particular has become quite a standout, and that's titin. I'd like to tell you a little bit more about these two different classes of proteins and remodeling structures as we go forward. So the way in which we know that these are definitive cardiomyopathy genes really is based on lots of familial disorders in which individuals who are known to have a genetic relationship, their sisters, brothers, aunts, cousins, have been clinically identified by routine imaging studies, sometimes associated with, unfortunately, sudden cardiac death events in which we can see the profound changes in cardiac morphology, and this enables the constructions of very large pedigrees. This led to the application of genetic linkage analyses, where we could scan through all of the chromosome locations, identify one that was implicated in the disease, and ultimately define the pathogenic mutation in a selected gene. The reason that we know this is so definitively true is because the statistical power of a very large family is really quite remarkable and allows you to predict how randomly such a co-inheritance would occur in all of the 40 people affected on this screen as compared to those who are not. And as you can imagine, that statistics shows that that would be extraordinarily unlikely to occur by chance. But think about where we are today. Today, instead of using that roving circle that takes several postdoctoral years to spin, we can go much more directly to actually sequencing not only exomes that encode all the protein sequences, but increasingly genomes, with one little box that can sit pretty much on your tabletop. And we know that this high capacity not only gives us the actual GATC sequence, but it allows us for the first time to really robustly interrogate whether the numbers of each of those base pairs are appropriately 2 copies, one for each of the autosomes, or if indeed there are copy number variants in genes, a new source of considerable kinds of human pathologies. If we have an individual whose exome or genome we choose to sequence from a pedigree such as this, and we find a very, very rare mutation by looking in dbGaP and other databases, 1,000 genomes and the like, we might have a fair degree of confidence that that might cause the disease. It becomes all the more powerful if we look at another distantly related individual who will share only 1/32 of his genome with his cousin over here, and that then makes it highly unlikely that these single rare variant that's identified in both of these individuals would have arisen by chance, sort of the exome equivalent to doing linkage analysis. But what happens when we don't have such a robust family history, or indeed no family history of disease, and we have a single individual patient who comes in? Can we make those same confident predictions that a variant that we identify in a gene, even one previously known to cause disease in the context of families, do we know that that really accounts for the disease in that individual? Well, I want to just briefly remind you that in addition to having variants that we inherit from our parents, we also have the opportunity to acquire our own personal de novo variants. And it's estimated now that there was approximately 1 rare de novo variant in each and every one of us. that about 1 in 50 of us will carry a pretty substantial copy number variant as well. And if we look not just at exome sequences, but indeed across the genome, we see that there are literally scores of mutations, new variants that have arisen in us. And the significance of these becomes obviously very, very difficult to interpret without family members and without knowing a whole lot more about the gene. So how do we take our ability to know genetic causes of familial disease then and bring it to populations and patients in the clinic? How can we discern the clinical significance of cardiac gene variants in each and every patient before us? Well, I would suggest to you that's going to require a very, very big database. So I hope that NCBI will get more funding, Dr. Collins. I think that certainly that's going to require a substantial harmonization of phenotypes so that we know when we say that this variant is found in this collection of phenotypes that we're all talking about exactly the same thing. And remember that a lot of our phenotypes are pretty nonspecific. A chest X-ray is probably seemed very simplistic, but it's not that very dissimilar from doing an echo or an MRI of a heart. It's not exactly a histologic analysis. Similarly, we're going to need to collect all of the definitive mutations and compare them to those variants in the same gene. We're going to have to add in what happens when you have different lifestyles, exposures, environments, and ultimately come up with some kind of integrated analyses. So in the first part of this talk, I'd like to tell you some of the strategies that we're trying to use to try and overcome these very big obstacles. And in the second part, hopefully convince you that it's worthwhile because we can learn some interesting biology. So when we think about the contractile apparatus, many of you may be remembering of a slide similar to this, in which you know that the motor proteins in all muscle cells are composed of the myosin heavy chain gene, which myosin heavy chain protein, that has to make contact with the thin filament and, like a ratchet, slide across it and create a power stroke. We've known for a long time that mutations in the 8 major components of the molecular motors of cardiac cells, the sarcomere, can give rise primarily to hypertrophic cardiomyopathy. These are from familial studies in which an autosomal dominant mutation segregates in a large pedigree, and we have robust statistics to say these are definitive disease genes. Well, we also know that cardiac hypertrophy, that remodeling I showed you on the first slide, is very commonly found in the general population. Indeed, 4% of the general population has unexplained cardiac hypertrophy, and it's largely attributed to diabetes, increase in blood pressure, and lots of other common cardiovascular disorders. Well, we thought it would be interesting to know if sarcomere variants contributed to that kind of remodeling. And to do that, we've capitalized on 2 different cohort studies. The Framingham Heart Study, which is right now a lovely community being buried in snow. And as all of you know, it's had about 50 years of longitudinal study of over thousands of subjects who have kindly participated in showing us lots of important pieces of information over more than 8 now different serial examinations. We've also looked at the Jackson Heart Study, in which there's less longitudinal information, but again, at least now 1.5 robust profiles in which we have cardiac imaging and lots of ancillary phenotypes as well. So when you take these 3,000 individuals and you say, let's just sequence all of those 8 major sarcomere genes that cause hypertrophic cardiomyopathy in these 2 cohorts, we had expected that we would find some variants and that would directly correlate with their left ventricular wall thickness or their degree of hypertrophy. Well, what we found was not some, but 11% of the general population have a change in the sequence that alters the protein of these 8 sarcomere genes amongst these 2 populations. Moreover, because since the discovery of these genes as causing hypertrophic cardiomyopathy, a lot of clinically robust criteria have been developed to actually discern which are the pathogenic ones and which are common variants. And so we applied those CLIA-approved classifications, and we could restrict those 8 rare variants down to about 0.6% of the population who would be deemed pathogenic if their blood was sent to one of these diagnostic laboratories. Well, that may not seem like a very big number to you, but that's about 3 times more prevalent than all given reports of the disease incidence is in the general population. which would make you conclude one of two things. One, we're miscalling variants, or two, we're missing a lot of the diagnostic criteria in the general population. And so when we looked in detail at the phenotypes of these variant carriers, we found that only about 1 in 10 of them had any criteria, even the softest call, of having phenotypic manifestation. And this is restricted to the Framingham cohort, where we have more than 60 years of longitudinal information. So it's not as if these people are likely to develop it over time. It really implies that we're not calling all of the variants in a correct manner. It doesn't mean, however, that those variants are completely silenced, because again, capitalizing on the longitudinal phenotypes, when we looked at adverse myocardial events that occurred in the variants carriers in comparison to those people in Framingham who did not carry the variants, we saw almost a 3-fold increase in risk of adverse events. And this is after accounting for the usual risk factors: smoking, diabetes, hypertension, and the like. So taken together, what I've tried to tell you is that we've known from decades now of research lots about highly penetrant monogenic pathogenic mutations, and that these occur in a cadre of genes that we can begin to study in the general population. When we do so, we find lots of rare variants in that population, but the penetrance and expressivity of these appears to be significantly lower. It's not absent, it's just significantly lower. So one of the big challenges then begins to say, what are the modifiers that are influencing the expressivity? Are they genetic? Are they background? Are they exposures? And can we use that information to actually take and modify the impact of these pathogenic mutations? That's a long way into the future right now, but I think it's something that's going to be important for us to begin to grapple with. I'm going to shift gears now and tell you a little bit about the other form of cardiac remodeling. And by doing so, I want to introduce what is becoming the most popular gene in my particular laboratory, Titin. Titin is not Titan, as some of my clinical colleagues will say. It is the largest gene and the largest protein in the human body, but it's a T-I-N, not Titan. And it is huge in terms of size. It actually is approximately 100,000 base pairs long for the protein encoding sequences. The molecule, as you can see, is almost 1 micrometer, and it spans 1/2 of the entire sarcomere. So this is one Titin molecule here. Here's another one overlapping here. As you can see from the descriptors below, Titin has been long known to be very critical in terms of a lot of capacities of sarcomere function. It's believed to participate in development and actually serving as a scaffold for the development of the sarcomere during embryogenesis. It clearly is important, not particularly in forced generation, but in forced transmission. And because it's been recognized that this purple domain here, designated M, the M band, has kinase activity, that it may also be involved in signaling. At the 5' end of the molecule, the Z band is the Z disc interacting region, another area that is known to be very critical for actually transducing signals. So several years ago now, we began to ask whether we could find causes of idiopathic dilated remodeling by looking at some small families, and increasingly, because there were not very large kindreds for analyses, to isolated patients. And we found that in those very severely affected patients with dilated cardiomyopathy and those rare families, that we could identify mutations in the giant protein Titin. And we then asked, since we've known that this is a single gene that is very big, that maybe we could also begin to glean strategies to interpret variants in single individuals by doing a deep dive of sequencing this giant molecule in large cohorts of patients. And so, as you can imagine, we took the same strategy I told you about before with a little of an exception. First of all, we took about 500 individuals with dilated cardiomyopathy who had been exquisitely phenotyped. This has been work done in collaboration with Stuart Cook at the Imperial College of London and now in Singapore. We've had expensive MRI and electrophysiology on these patients, which has helped to inform some of the phenotypic manifestations of these mutations. We've again sequenced both Framingham and Jackson, over 3,000 individuals, and because we know that there's a very rare form of pediatric onset dilated cardiomyopathy, we had a chance to look at a small subset of patients with these, about 50 in total. So what we did is to do robust sequencing using an Illumina platform and use a standard pipeline for analyses. And suffice it to say that when we used this and then began variant calling, we had the unfortunate circumstance of recognizing that the number of variants in this gene alone are absolutely astronomical. There is one missense variant that is rare, not identified in the vast majority of databases, or at least occurring in less than 1% of the population in each and every one of us. Without a family history, without the capacity to deeply examine the biophysical consequences of each one of those, there are going to be a lot of variants that are of unknown significance in this gene for a very long time ahead. And so what we've done is to restrict our analyses to those variants that appeared to have a more profound capacity to change what Titin does. And those are the ones that actually prematurely terminate the encoded protein, either because of a premature stop sequence, a nonsense, or an essential splice site mutation that result in frame shifts and alternative splicing. So when we employed only the analyses on those truncating mutations, we found a very interesting result as shown here. For those patients with end-stage, really profound dilated cardiomyopathy and heart failure, these are individuals who have been referred for transplantation or who are undergoing VAD treatment, ventricular assist device treatment, we saw a significant enrichment in the Titin truncation mutations found in that population as compared to healthy volunteers and Framingham and Jackson. So too, those individuals who had dilated cardiomyopathy by echocardiography, by MRIs, but were not yet progressed to heart failure, they were also significantly enriched in comparison to this other population. But, as you'll see, there were not zero pathogenic sounding or looking mutations in the general population. They were certainly present, albeit somewhere between 1% to 3%. Well, that may not seem like a very high percentage, but if it was your heart, I think that some of us might indeed be worried about it. So we began to ask whether there were other strategies that we could use to try and tease out which are the meaningful variants and which are not. And the first thing that we did was to try and ask whether there was a distribution of these variants along the protein that might be informative. And shown here on the top are the ambulatory, the severe DCM patients, and the Jackson and Framing heart, as well as, I didn't include this, normal controls, distributed along this region. And it's not a very impressive clustering, I would suggest to you. And so the next thing that we asked was whether there were differences in the location of these variants, not along the longitudinal distribution of this large molecule, but the degree with which the exons in each of those domains are expressed. Like many very great proteins with multifactorial capacities to perform different processes, Titin undergoes lots of different alternative exon usage. And so we then restricted our analysis to ask which are the exons that are highly expressed in the heart as compared to those that are lowly expressed. So this is a region of expression based on human heart tissue. The higher the gray peak, the higher the level of expression. And what you'll see is that along this region here, there's considerable dropout because these exons have much, much less expression within the human heart. You'll also note that a lot of the variants that are not found in our dilated populations reside in that region. And so if we eliminate those that are expressed and truncating from only the lowly expressed exon, you get a far more clustering kind of approach with substantial enrichment along the domain that is called the A band, a portion of the molecule that we know very little about in terms of its function, and that is significantly enriched among those with DCM as compared to the general population, whereas those that are enriched in the general population turned out to be in these lowly expressed exons. So this is an idiopathic dilated heart in which there is a global ventricular decrease in function, but there is no regional wall abnormality, nothing particularly remarkable about the intensity of the image quality around here, and the histopathology is what I like to call quite vanilla. It doesn't tell you either the etiology or whether this is going to promote arrhythmias in a patient from whom it came or not. And so we asked whether knowing this TTN mutation would actually inform phenotype outcomes in patients, again, based on the detailed information that we had about them. And so from our patients with MRI, we looked at those who had the mutation, shown here, versus those that did not, and you see a slight decrease in left ventricular ejection fraction, a measure of contractility in those that were mutation positive. But perhaps more impressive to me was the fact that their wall thickness was substantially decreased in comparison to other forms of dilated cardiomyopathy. And sadly, the number of arrhythmias that they had, significant ventricular arrhythmias, were substantially increased. So this has provided to us the first time that we're beginning to tease out a phenotype that may be genotype selective in the dilated population. Moreover, I told you before that we had the sense from the distribution of mutations in exons that are highly expressed, and in particular in that A domain clustering, that those were more likely to occur in individuals who had the severe forms of dilated cardiomyopathy. Not surprising, that distribution also implied that they would have the poorest ejection fraction. The further the truncation occurred towards the 3' end of the molecule, the worse the ventricular function. That was true if you had dilated cardiomyopathy or if you were a Framingham heart cohort subject and simply carried one of these mutations, so that you actually had a decrease in your ejection fraction in comparison to those who did not. And it was worse the more 3' to the gene or to the more carboxyl end of the molecule that you became. I'm going to just for a moment tell you we've done those same kind of analyses in children who have pediatric onset of dilated cardiomyopathy before the age of 16. This is a devastating disorder that often, unfortunately, leads to rapid death if they haven't undergone cardiac transplantation. We've now looked at about 84 genes that have been identified from family studies in this patient cohort, and suffice it to say that a few mutations have been identified, not very many. There's a lot more to do in this cohort of patients. But most importantly, we see very few mutations within TTN, these truncating mutations, significantly less than we do within the adult population. So taken together, I think what this information has begun to tease out in terms of information that is relevant to interpreting TTN variants is that most of these appear to truncate the peptide, the pathogenic ones, that they occur in highly expressed exons within the left ventricle of the human myocardium. They are more regionally located than randomly distributed across the molecule, and those that are clustered towards the more carboxyl end, the A band of the molecule, are clearly associated with more severe disease, poorer left ventricular ejection fraction, greater risk of arrhythmias, and they are unlikely, if you find them in a pediatric onset dilated cardiomyopathy patient, to be etiologic. So if we can understand these kinds of clues that help us to recognize the pathogenic mutation, the next question, I think, is how are we going to deal with that information? How are we going to treat them? And obviously, the first thing to do is to understand mechanism. So when we think about a polypeptide that is truncated or foreshortened, we often assume that that means that it has a diminished capacity to participate in its usual roles. That may even be so profound that it is not incorporated and that we actually have loss of one of the functional gene copies of a molecule, or haploinsufficiency. So if we had haploinsufficiency as the mechanism by which TTN truncation caused dilated cardiomyopathy, we would not expect a selected distribution of DCM mutations arising at the 3' end of the molecule. Rather, they should be able to truncate the molecule anywhere along this huge polypeptide chain and equally cause disease. I've tried to convince you that those at the amino terminus appear to be far less devastating than those at the carboxyl terminus. So that raises the chance that this is not, in fact, due to haploinsufficiency, but rather it suggests something else. And that, again, has a lot to do with the giant size of this peptide. This is a very, very large transcript. And chances are the 5' sequences are going to be translated and actually begin to incorporate it into the sarcomere even before all of the 3' sequences have been generated into peptide. Moreover, we know that from very rare families where there are truncation mutations at the very M band, the most carboxyl end of the protein, that immunohistochemical studies reveal that those proteins are in fact incorporated into the sarcomere. So this is a profile of these kinds of patients. Control is shown in the first panel, and these are 3 individuals who have homozygous truncations in the M band region of Titin. And as you can see, antibodies that are selective for the Z-disc region of Titin, the A-band region of Titin, show robust expression even in those that carry these homozygous mutations, suggesting that these peptides are sufficiently stable that they actually are incorporated. I think this provides the most profound evidence that these are likely to operate by a dominant negative mechanism. So if they're dominant negative, what do? Well, we haven't had a chance to do a whole lot of work on this yet. iPS cells are being made, so is a mouse. But I can tell you that some of the hypotheses we can put to rest, namely that they might disrupt the formation of normal sarcomere structure. We've now done lots of electron microscopy on human hearts that carry these mutations, and as best we can see, sarcomere structure is completely normal despite having these pathogenic mutations. What we do see is lots of interesting changes in nuclear morphology, something that hints that there might be significant abnormalities in mechanosensation and stretch and passive force on the nuclear membrane. And these rather bizarre nuclei are pretty profound in comparison to what we see in other causes of idiopathic dilated cardiomyopathy. While we know that a lot more work will have to be done to try and build robust models to do this, I want to outline for you how we could use this information that a truncation of Titin that is acting in a dominant negative mode of action might provide a new way for treating. The first thing is obviously to develop models in which we can robustly look for haploinsufficiency versus dominant negative. We can do all sorts of transcriptional profiling to identify the signals that might be perturbed, in particular because of the disruption of the M-band kinase domain, to build appropriate networks, and then to identify candidate molecules, which we would introduce into isolated myocytes and the like, see how they perturb cardiac biology and physiology, and then see if any of these things might play out as potentially beneficial to our patients. We've done a lot of this already in the mutations that cause hypertrophic cardiomyopathy, in large measure because we've been aware of the biophysical consequences and signaling properties of those for several years now. We've made the mice and done the studies, and I just want to tell you a little bit about what those studies have informed us. I want to remind you by telling you these are in the contractile apparatus that are really generating the sarcomere power and force because they occur in myosin, actin, the thin and thick filaments as well. And they result in a very remarkable histopathology that's quite different from dilated cardiomyopathy. The myocytes are enlarged in size. They're separated by interstitial material that, with appropriate staining, is a robust increase in fibrosis and extracellular matrix within the myocyte. This kind of dysmorphology ultimately results in diastolic dysfunction and produces the symptoms that we see in patients with hypertrophic cardiomyopathy. How could a myosin mutation do this? Well, the first thing we need to understand is what does the myosin mutation do to sarcomere performance? With collaborations in David Warshaw at the University of Vermont and more recently with Jim Spudich at Stanford, we've been able to engineer human mutations either into cells and have them robustly expressed in vitro, or indeed from mouse models in which we can isolate the mutant from those animal hearts. And what we've seen is that, quite surprisingly, mutations that result in hypertrophic cardiomyopathy do not decrease the capacity of myosin to perform. Rather, in fact, it increases it. Shown here is a sliding velocity of myosin along actin and force generation in a wild type compared to hypertrophy mutation 403, one of the more profound causes of hypertrophic remodeling. We see that there is increase in force production. There's actually increase in sliding velocity along actin and ATPase as well. Another mutation shown here has somewhat different distributions in which of those different biophysical properties are perturbed, but both of these will do exactly the same thing. They will increase the power of the contractile apparatus, not decrease it. Well, if that's the case, if we are increasing power, how are we producing those histopathologic changes that we see? What we think is going on right now is from the increase in power from the sarcomere gene mutations that we are resulting in an increase of signaling that is transmitted to the non-myocyte population, the non-myocyte being the fibroblasts, the cardiac fibroblasts that are resident within all of our heart cells, and that the biophysical mechanics are actually transducing signals in those non-myocyte cells. This is interesting because these non-myocyte cells, the fibroblasts, don't even contain the mutation. But the activation of signaling on them is so powerful that we see increases in calcium homeostasis, increases in TGF-beta signaling that leads to a profound increase in the proliferative capacity of these cells, the deposition of periostin, connective tissue, and the like, and that over time results in deposition of the extracellular matrix that we see in human hearts. We can challenge this kind of pathway with lots of pharmacologic interventions, either those that decrease the mechanical power or more directly those that inhibit TGF-beta signaling, either with antibodies or with some of the pharmacologic agents. And when we do so, we're able to diminish the amount of extracellular remodeling that occurs in the hearts with a hypertrophic mutation. Well, that's well and good, but that's a secondary consequence to the actual deficit or problem of the heart, namely the myosin mutation itself. And so one of the things we've asked is whether we could harness newer technologies to really grapple with the closer proximal problem, the actual gene mutation. Whether we could, in fact, silence the myosin mutation that is one of two copies within these animals. And to do that, we've employed a system of adeno-associated viruses, in particular serotype 9, which is known to be cardiotropic. We've manipulated that a bit so that we have it driven under the expression of a cardiac troponin T so that there is exquisite high-level expression within myocytes. When we first drive an EGFP vector, what we can see is we can generate lovely green hearts that are not resulting in increase of EGFP expression in other tissues. And I'll tell you that that lasts for at least 5 months period of time after a single injection of the AAV virus on day 1 of birth. If now, instead of just having a nonspecific vector such as green fluorescent protein, we use RNAi, we thought this might provide us some capacity to decrease mutant expression. Well, if we're going to do that, we'd have to be pretty careful because this is a dominant mutation. There's a single nucleotide change in one of 2 alleles and would not be beneficial to the mouse and certainly not to humans if we knocked out both copies of myosin expression. We know that that's a lethal phenotype from knockout mice. So what we did initially was to ask whether we could selectively knock down the mutant expression with shRNA that in particular tagged the particular mutation, and whether that would be specific to discriminate between mutant and wild type in cells. When we did that, as you can see, in comparison to shRNA that is scrambled here, there's no change in mutant and wild type expression, that our quote-unquote mutant selective shRNA was woefully inadequate to discriminate the mutant from the wild type. Now this is, again, different from the wild type allele by a single nucleotide. So if we add one more variant in the shRNA, so that we are now 2 nucleotides different from wild type, and only one nucleotide different from the mutant allele, we achieved a remarkable different result, with suppression to about 25% of the prior level of mutant RNA expression. Is that sufficient to do much? Well, we injected these shRNAs into our hearts, into our mice rather, and evaluated them approximately 8 weeks after the injection, and the result was truly astonishing. Recall now that we're reducing at most by 25% the amount of mutant RNA expression. And this is a series of cardiac evaluations in our mouse hearts. Here's the wild type, and what you can see is the left ventricular wall thickness of a mouse heart is about 0.7 millimeters. And in a hypertrophic mouse, because it has the 403 mutation, it's about twice that. If we reduce the level of expression of the mutant allele considerably, we get it back down to normal. Well, that's a nice nonspecific phenotype. The immunohistochemistry and analyses are far more impressive, and I show you them here. Shown here is an animal that was treated with scrambled shRNA. You see the interstitial fibrosis in this region of the myocardial section here, and I hope you can see that there's disarray and accumulation of fibrosis by histopathology sections. In the animal treated after 8 weeks with the shRNA, we see none of that. This is undistinguishable from a wild-type animal. Similarly, the kinds of increased expression of markers of hypertrophy, such as the A and F gene, are enormously reduced when we have maximum viral delivery, 5 × 10^13 viral particles per kilogram. The amount of fibrosis is similarly reduced. Again, for the clinicians in the audience, we know that when you have a hypertrophic remodeling, you have lots of EKG changes, and we also see those in our mouse hearts. Shown here is a widening of a QRS complex that occurs quite commonly in the hypertrophic mouse. We can normalize that to its normal dimensions of 14 milliseconds after treatment. So, taken together, I've told you that we can now begin to know some of the mutations that are definitive causes of cardiomyopathy. We can begin to say that these are likely to be acting through a dominant mechanism, not because of haploinsufficiency, and that we have a strategy to potentially silence them in an allele-specific way in mice. Could we bring this to the human population? I wish it was Friday afternoon, because in Boston, that's a time when the labs all gather together and have a beer and enjoy each other's company, and it makes you dream a little bit. And we cardiologists are sometimes very big dreamers. We're not afraid to tackle things. What I'm showing here in this schizophrenic echocardiogram is an image of a human heart in which the head of the individual is here, their feet is over here. The echo is showing the blood going from the left atrium to the left ventricle, and this big fist area over here is a highly thickened interventricular septum. It accounts for enormous amounts of symptoms and can increase the risk of sudden death in individuals who have profound hypertrophy in that afflotract area. We know that this is so profound that we take 2 heroic measures to get rid of it. The first is to do open heart surgery and to remove that hypertrophied septum, what's called a myectomy. The less invasive way, shown here in diagram, is to pass a catheter into the aorta from the leg and retrograde to cannulate the vessels that specifically feed the interventricular septum. That's the one shown right here. That's called the interventricular septal perforating artery, and to inject alcohol in it so that we have given the individual a heart attack to have that hypertrophied septum die. I would suggest to you that I don't think it's too far off to think about, rather than killing the myocardium in that region of the heart, to use these kind of approaches to potentially silence mutations. If we could achieve long-term silencing of even 25 to 30%, we might substantially reduce the hypertrophy in that region and spare the myocardium of death. Now, those of you who are real human geneticists are saying, well, that's great, you can do it for one mutation. How many people have that particular mutation? And you'd be right. Private mutations are the name of the game in lots of important diseases, including all of the cardiomyopathies. There are now more than 1,000 different mutations that have been identified to cause hypertrophic cardiomyopathy. There will be an equal number that cause dilated cardiomyopathy. And the capacity to design such a strategy for each and every one would be profoundly limited. But what we can do is capitalize on the extensive knowledge that's come from human genome sequencing and to begin to ask whether we could, instead of targeting the precise mutation, to target a nearby variant that is in linkage dissociation with that mutation. It's on the same allele. It actually is co-inherited with the mutation. And to see if that was even feasible, we went to a favorite source of ours, the NHLBI exome variant server, looking at the myosin heavy chain gene. And we can see here that there's a variant, but this one turns out to be in an intron, which would be hard to silence from an RNA point of view. But here's a coding synonymous variant in one intron. And here's another one over here. So if we were to now test that strategy, we can again do this in our mice by taking our inbred mouse, breed it into a different background, and now try and silence not the mutation, but the nearby polymorphic SNP. And when we do so, the results are essentially the same. So taken together, what I've tried to tell you is that we think by actually considering the use of nearby SNPs that are tightly linked to mutant alleles, that we could build a battery of approaches to silence mutations in a very specific way that would leave the non-mutated allele to make good protein. And while it would be unlikely that we could completely reduce the mutant level to zero, that we might have the capacity to reduce it significantly to attenuate the remodeling that occurs. So where are we going with all this? Well, I hope I've convinced you that there are lots of cardiovascular disease genes. They've been powerfully important in teaching us about sarcomere function and sarcomere biology. I think that they're increasingly going to become very useful in understanding phenotypic variation in the clinical setting. We know that we can do gene-based diagnosis. There's a lot more for us to be able to do that in a very robust fashion. But with the databases that are being developed and with the increased use of genetic testing, I'm absolutely certain that we'll get there. Moreover, shRNA is only the beginning of trying to silence mutations in somatic tissues. By CRISPRs, most recently, I think there's enormous capacity to use genetic antidotes for treating not only heart disease, but a variety of other conditions. And I would encourage all of you young people in the audience to think about getting involved in this really, really exciting area of science. I'm going to stop there. I simply have to acknowledge this amazing team that I work with. In particular, Alex Bick, Dan Herman, Paul Lakeet, and Karua, who are basic researchers in the laboratory and also MD, PhD students, my clinical colleagues, Carolyn Ho, a whole team of sequencing colleagues from the Broad, from the Jackson and Framingham Heart cohorts, and my colleague from the UK, Stuart Cook. Thank you very much. I'd be happy to answer any questions.
Cricket, that was terrific. Yes, please, people do use the microphones to pose questions so people listening on the web can hear as well. Please go ahead.
Cricket, that was great. Thank you very much. And when those MD, PhD students are ready for our Alaska program, remember to send them this way.
So I will.
This is a question about your My6403 mutant where you used shRNA. I hope I got that mutant right. And the link to alcohol ablation of the septal hypertrophy. Have you, in the mice, allowed them to develop the hypertrophy and then see if you could reverse it using that shRNA strategy?
Right. So the question is, can we reverse established hypertrophy using the shRNA approach right now? Those are experiments that we have done only in a short-term sequence right now. We have not done them in a full 8-week to 12-week period of time. And we think that those are important to do for a protracted period of time. When we do a septal ablation, it takes months for the myocardium to remodel, and that's after killing the heart muscle. So we would anticipate that it will equally take many months to remodel a hypertrophied mouse heart if it has the capacity to do so. Do we think it'll work? Yeah, I think it will. There's turnover of myosin proteins in all myosin tissues, so we would expect that the increased amount of wild-type alleles should allow for that increased force to be diminished over time. And the second reason I think that is anybody who's treated a patient with hypertension and hypertrophy, it takes time, but over time that left ventricular hypertrophy will resolve. So we're hopefully will happen. We just don't have the results yet.
I have a basic question about the Titan part of your talk. In an individual who has a heterozygous truncating mutation, are the two alleles translated in 50-50 proportion, or is the truncated allele somehow have an advantage because it can be translated faster?
That's a very good question. We've begun to look at that the one good news about Titan is there are so many variants in there that you can look to see from RNA-seq analysis if both in fact are expressed. And we've gone ahead and looked at that, and we see no increase nor decrease in allelic specific SNPs as we march along the Titan molecule. So we don't see an advantage nor a disadvantage.
In the patient, you had alcohol infusion. How long it was effective, and what is the outcome in terms of cardiac abnormalities by MRI or echocardiography?
Right. So alcohol septal ablation is, just so we're clear, not something that I routinely do, but many of my colleagues do. It is usually recommended for individuals who have, first and foremost, a lot of symptoms that have not responded to pharmacologic interventions. And for reasons that the patient often is involved in, or because there are clinical indications, that person is not appropriate for referral to surgery, to open heart surgery. It's increasingly being used because of patient preference. It usually takes a period of 6 weeks or longer, 6 weeks to 3 months, to see remodeling and regression of the interventricular septum so that the outflow tract becomes more opened. And from the long-term follow-up, we do not see it growing back. Whether that will happen in 5 years, I don't know, but right now we have not seen it.
You're not thinking of ligation because that will be more devastating, I guess, rather than this chronic effect of alcohol. Is it one-time use alcohol, or?
I'm sorry, is it?
One-time
Is it
use.
one?
Oh, it's one-time use. It's one-time use. One-time use. And I will mention for people who are not interested in this clinically, the advantage of this AAV thing is it's also one-time use. And you get 5 months of silencing the gene of your choice from a one-time injection in a mouse. That's a pretty good experimental system, which we are using for lots of other investigations.
Thank you. More part.
Just wondering about the Titan part of the talk. Is the differential expression of seen in the Titan gene, is that thought to be protective? And have you delved a little bit into what genetic sequences might be contributing to that?
I'm sorry, is the...
So within the Titan gene, you mentioned you showed different expression patterns, so that different parts of it seem to be expressed more than others, and that there's a correlation with regards to the location of the mutations compared to the expression patterns. I'm just wondering if that expression pattern difference is hypothesized to be protective.
Right. So the regions where the normal population have accumulated some variants that truncate the molecule are in parts, can be involved in making Titan molecules that are at very, very low level expression. So they're alternative exons that are spliced in and spliced out because there's a lot of different Titans in each and every heart. But the abundance of them is very, very low. And indeed, the ones that are incorporated into the sarcomere, almost by definition, because they span from the M band of the sarcomere to the Z disc of the sarcomere, are what we call full-length Titan or full-length sarcomere Titan, it's referred to. And it does not include those lowly expressed exons. There's also another, for those aficionados, something called the Novex3 isoform, which is a very, very short 5,000 base pair transcript, and no one actually knows what that does. If it does anything.
Cricket, the whole Titan story gets more interesting all the time, and I guess you're really causing many of us who depend upon nonsense-mediated decay to predict the consequence of a nonsense or a frameshift mutation, to question whether we should depend on that if the gene is anything other than, hemoglobin, which is kind of short. Because it sounds as if, maybe because of the coupling of translation,
right,
transcription, splicing, that you already passed the point of not having a protein before you hit that stop codon. I assume, though, that there would be some relationship, you were talking about this a minute ago, in terms of the abundance of the mutant allele depending on where the truncating mutation was, and that if it was fairly early on, you would get somewhat reduced amounts of that transcript by nonsense-mediated decay, whereas if it's very near the 3' end, almost no effect at all. Now, part of the question I want to ask is, though, what is the phenotype of a true haploinsufficient Titan allele? And if it's not bad, does it say that what you're doing for these hypertrophic cardiomyopathies might also work for Titan mutations that cause dilated cardiomyopathies, where, again, you're just trying to silence an allele that's actually causing toxicity?
Right. So the most definitive evidence, I think, of a human patient, not a mouse, with a Titan haploinsufficient mutation, a null, has come from some rare recessive families, but it's not a null, I beg your pardon, it's at the 3' end. It's those M-band mutations. There has been no report that I'm aware of a homozygous or a heterozygous loss of function with overt disease.
So it might be wild type.
At the very, very amino-terminal sequence. That's correct, that's correct. And when we see where we find those clustered in our random people, they're more likely to be at that amino terminus. So there may be no abnormality of the heterozygote loss of function. When we do shift to mice now, and there is one complex mouse model that was a knockout, but it also introduced a simultaneous mutation. So it's a complex rearrangement. But suffice it to say that it appears that the homozygous is lethal, okay? The heterozygote is fine. So we do think that if it is a dominant negative mode of action, that silencing Titan would be the mutant allele would be beneficial.
And you would have plenty of ways to do that, given that Titan has such a huge number of variants that almost everybody's going to be heterozygous for something that's in the part of the transcript that's upstream of the truncation.
And we would like to have those that are common, so we didn't need to make 1,000 of them, just a couple hundred.
One more question.
You addressed it at the beginning, but what is the percentage of cardiomyopathies that would be attributable to genetics, and what is the percentage that would be attributable to lifestyle or environmental factors, or what is that?
Or all of the above, right? So that depends on the phenotype. So if you take individuals who have one family member who is also affected with a hypertrophic phenotype, 75% of those are genetic, okay? So that's a pretty high percentage. If you take individuals who have no family history of cardiac hypertrophy, and they have all of the classic manifestations of hypertrophic cardiomyopathy but no family history, 35% of those are genetic at tops, If we do the same experiment now with dilated cardiomyopathy, unfortunately, the best, even in a familial case that we're doing, is about 50%. And that's including Titan, where that's 25%. That's the biggest piece of the pie right now. And everything else is 1,000 other little genes. So there's still more to discover there. And when you take an isolated individual with idiopathic dilated cardiomyopathy and include Titan, we're at about 15%.
Well, Dr. Seidman would be willing to continue this conversation and a reception in the library. Please join for coffee, cookies, and that ongoing chat. And meanwhile, please join me in thanking our speaker again. Thanks.